of deoxyribonucleic acid, DNA, that make us who and what we are. When we talk about genetics, whether it's regarding microbes or us, it's important to remember that biology is not destiny. That is, who and what we are depends on what genes we have, but it's also important to remember that genes interact with the environment in terms of who and what we are. So there is a whole other field called epigenetics, which talks about the interplay of our DNA and the environment. However, herein, we'll just be talking about DNA strictly. Okay, when we say gene, what we mean is a length of DNA that encodes the instructions for making RNA. It's important to remember that we don't use the instructions in DNA directly whenever we want to make, say, a protein, for instance, which is encoded by a type of RNA called mRNA, messenger RNA. Instead, we make a cheap knockoff, if you would, of the DNA in RNA. It's very much as though one had gotten a vinyl recording of, say, a rare piece of music, and rather than playing that vinyl every time we wanted to listen to the music and risking scratching it, that is corrupting the instructions, instead what we would do is make, say, a tape. Okay, and in that case, if the tape were ruined, it wouldn't really matter because we still have the original instructions intact in the form of the vinyl. Okay, so we've said before that RNA is essentially the mouthpiece of the DNA. When we say DNA, what we mean is a polymer of deoxyribonucleotides that are linked together. Okay, deoxyribonucleotides, we mean deoxyribonucleotide triphosphates are serving as the monomer for our DNA, as well as the power source for forming the phosphodiester bond that links them together. Remember, deoxyribonucleotide triphosphates have high energy phosphate bonds that we can use for powering the dehydration synthesis used to link them together to form deoxyribonucleic acid, DNA. DNA for cells is found on structures referred to as chromosomes. The chromosomes carry the hereditary information, genes, that are necessary to make new cells, as well as operate existing cells. Up a level from that would be where we talk about genomes. Genome would be the entire genetic complement of an organism. So for instance, if we're looking at a prokaryote, like E. coli, we would say the chromosomal DNA would be that DNA found in the nucleoid. Okay, we can also have extrachromosomal DNA, which is not essential to the heredity or running of the cell, in the form of small circular pieces called plasmids. If we're talking about eukaryotes like us, particularly animal cells, then we would say that the genome consists of the nuclear DNA, which contains all of the instructions for making new cells, as well as extrachromosomal DNA that could be found in mitochondria, which we have as animals, as well as chloroplasts, which we would have if we were photosynthetic eukaryotes. As we said earlier, the prokaryotic genome, and that's cells such as E. coli, is contained in two structures. We have the large circular chromosome that's contained in the nucleoid. Okay, and the nucleoid appears to be electron dense. What we're looking at in this slide are electron micrographs. It's electron dense because in order for the DNA to fit into the cell, it has to be highly compacted. In addition to the DNA that's in the nucleoid, that is the chromosomal DNA, we also have the proteins and the RNAs that are associated with it. Okay, if you look on the right panel, what we see is a cell that has had all of the DNA extruded from it, much like that craft called eggery, where one blows out the contents of an egg so that one can use it, the shell that is, for crafts. And all of that spaghetti-like material is the bacterial chromosome. Okay, the E. coli chromosome, for instance, is 4.6 million base pairs, okay, long, and that would be 800 times longer than the cell. So again, it has to be compacted so that it will fit in the cell, and this is what forms our nucleoid. Okay, in addition to the chromosome, in prokaryotic cells, we can also have small circular molecules of DNA, that is plasmids, and these are separate from the chromosome. Okay, the chromosome is the main portion of the DNA. That is the DNA that is essential for running the cell and also for being put into new cells. That is the DNA that we need to make new cells. Okay. And that DNA has associated proteins, such as the proteins that are involved in replication, that is making new DNA for cell division, as well as proteins that are used to repair DNA if there's damage. Also, remember that we make RNA using the instructions of DNA, so the nucleoid would also contain the RNAs that are being produced using the instructions in the DNA. Prokaryotic cells have a single copy of each chromosome. That is, they're haploid. So, for instance, if we were to damage a gene on the chromosome, there is no other copy. There's only that one copy. And so this makes prokaryotes, for instance, more amenable to genetic studies because we only have to worry about one copy of each gene. Plasmids. Plasmids are small extrachromosomal molecules of DNA that replicate independently. This does not mean that we can take them out of a cell and get them to make more copies of themselves. What it means is that regardless of what the chromosomal DNA in the nucleoid is doing, the plasmids can go on having more copies of themselves made. So, for instance, there can be more than one copy of a plasmid per cell. Plasmids contain the information that's required for their own replication, and then often they will have information for one or more cellular traits, such as antibiotic resistance. It's important to remember that plasmids are not essential for normal bacterial metabolism, for growth, or for reproduction. That is, one can look at plasmids as bonus DNA, much like the bonus room that many of us may have as part of our homes. That is, the room that used to be a garage, but now has been turned into a family room. We don't need to have a family room, but it's nice. Bacteria don't need to have plasmids. However, it can be nice in that, for instance, an antibiotic resistance plasmid can confer survival advantages. That is, they can contain information that cells can use to survive in the presence of antibiotics. Regarding plasmids, there can be a number of different types of plasmids, each of which has a distinctive property that they can confer on the cell that has them. Remember, plasmids are not essential genetic information. Okay, so we can have fertility factors, F, also called F factors or F plasmids. These contain the information that cells use to make conjugation pili, which they can then use to share genetic information among other members of their population. We can also have resistance, R factors, or R plasmids, which have genetic information that can be used for making proteins that allows cells, for instance, to destroy antibiotics. An example would be an R plasmid that contains the genes for beta-lactamase. Beta-lactamase is an enzyme that can degrade penicillin, and therefore cells that have a resistance factor that contains a beta-lactamase gene would be resistant to penicillin. So they would have an advantage if they were growing in an environment such as a hospital setting where penicillin is being used. We have, thirdly, bacteriocin factors. So, for instance, bacteriocins are proteins typically that cells can use to kill other typically closely related cells. So for instance, E. coli can make colicins if they have a bacteriocin factor. And the idea is that by being able to kill cells nearby, that allows them to gain a bigger share of the nutrients and available space in an environment. Virulence refers to how sick, how diseased an organism can make us. So pathogenicity refers to whether or not an organism can make us sick. Virulence refers to how sick it can make us. So for instance, salmonella can have virulence plasmids that encode proteins that are used to modulate the immune system to prevent the salmonella being killed. That is, they are used by the salmonella to turn off or turn down the immune system. And if you know, for instance, salmonella really doesn't stimulate the immune system very well, so that one can get salmonella, that is food poisoning, bacterial food poisoning, time and time again because there's no immunological memory to it. In contrast to prokaryotes, which have circular chromosomes and a nucleoid, something like a nucleus but not a nucleus because there is no surrounding membrane, eukaryotes have chromosomes that are, one, linear, not circular, and that are contained, sequestered within a membrane-bound structure, the nucleus. Typically with eukaryotes, there will be more than one chromosome per cell, and typically chromosomes come in pairs. So eukaryotic cells will have two copies of each chromosome. Therefore, we say that they are diploid. Because eukaryotes have two copies of each chromosome, they would have two copies of each gene, again, diploid. What we're looking at here is what's called a karyotype. That is a rendering of the chromosomes at a particular point in the cell cycle when they've duplicated, and some clever person has figured out a way to color each chromosome characteristically so that, for instance, chromosome 1 is gray, chromosome 2, those are kind of a fuchsia purple color, and so on. This particular karyotype is that of a male. So we would have 22 autosomal chromosomes. Okay, that is chromosomes 1 through 22. And then we would have a 23rd pair that are the sex chromosomes, the X and the Y. XY is male, XX is female. When we talk about eukaryotic chromosomes, we have to talk about packing, because like the case with prokaryotes, the DNA, the amount of it, is too long to be contained in the nucleus if we don't have a way of compressing it. So the initial compression comes in the form of nucleosomes. Okay, and so we would take the DNA and then wrap it around cubes of histone proteins to form a nucleosome, and then those would be linked by short stretches of DNA with histones. The short stretches are called linker DNA. This looks like beads on a string and is in panel A. In panel B, we've further compressed the DNA with nucleosomes, which is called chromosome or chromatin, into a chromatin fiber. And then in panel C, what we do is we take the chromatin fiber and form it in a series of loops. We can have loose loops, which is loosely packed, as we see above. That would be active DNA. That is, the genes in those regions can be gotten at by the machinery that's needed to make RNA from them. So those genes would be said to be on. So for example, if we were thinking about an eyeball cell, then the genes that are involved in making a cell decide to be an eyeball cell would be in the active or loosely packed chromatin. On the other hand, okay, if there were genes that were, say, involved in making liver cells, then we don't need those in an eyeball cell, so we would put them in the tightly packed, the very densely looped chromatin, and now the machinery for making RNA can't access that area, and so these would be inactive. That is, the genes are turned off. So this is a way of regulating gene activity and determining what cells become what. Finally, in panel D, we get to chromatin that has been highly condensed, as we would need to have when we have cells dividing. So this is typically what we think of when we think of eukaryotic chromosomes. To go back a little bit to panel C, we talk about euchromatin. That would be the loosely packed chromatin. Heterochromatin or other chromatin would be the tightly packed chromatin. As we noted earlier, in eukaryotes, we have both the chromosomal DNA that's found in the nucleus, but we also have extranuclear DNA, that is DNA that's not found in the nucleus. For us, as well as, that is, animals, as well as certain other cell types, that DNA would be found in mitochondria and chloroplasts. And if you recall our lectures on cell structure, mitochondria and chloroplasts have circular DNA like prokaryotes because we believe, according to endosymbiotic theory, that they arose from prokaryotes. Now, in the case of this DNA, it only encodes about 5% of the RNA and proteins made by the cells. Okay, this is nowhere near the complement of DNA that the prokaryotic ancestors of mitochondria and chloroplasts had. This is why we can't, for instance, take mitochondria and chloroplasts out of the cell and have them divide in, say, a petri dish with the right nutrient media. They don't have all of the genes that they need for maintaining themselves. Those genes have migrated over time to the nuclear DNA, which codes for 95% of the RNA and proteins. Some fungi and protists, notably some protozoa, can have extranuclear DNA in the form of plasmids. However, again, we, animal cells, cannot do that. Another thing that we need to think about when we're talking about gene function is the difference between genotype and phenotype. Genotype would be the set of genes in the genome. That is, we have, looking at the cross below, we have one parent that has the genotype big R, big R, okay, and so it can only make gametes that are big R. We have the other parent, which has white flowers. That one's little r, okay, and so it can only make gametes, sex cells, that are little r. So the genotype of one parent is big R, big R. The genotype of the other parent is little r, little r. Okay, now when we talk about phenotype, okay, then we're talking about the physical features and functional traits, that is metabolism or appearance of the organism. So when we do this cross, we're going to cross a gamete that's big R and one that's little r. All of the progeny will have the genotype big R, little r, okay, but the progeny will all have the phenotype of red flowers. Okay, so we had the one parent that was genotype big R, phenotype red. We had the other parent that was little r, phenotype white. Okay, they were both considered to be homozygous. That is, they have two copies of each gene that are the same. Okay, then we cross them. We get the genotype big R, little r in the progeny. Okay, and so that would be the genotype, which would be considered heterozygous because we have different forms of the flower color, the R gene, and then we have the phenotype where all the progeny, despite having a little r gene, okay, are red.